form new structures and new shapes. We need to think about differentiation, and we need to think about growth as well. I'll start giving a kind of broad overview today, and then we'll pick apart all of these individual things as the next subsequent lectures take shape. Okay, so here I realized last week that I had just jumped into particular terms that may not be so familiar to all of you. So fate, for example, we all know what fate is. We're Indians, we live on fate. But here we're thinking about a cell's fate, and it's what it becomes during normal development. Okay, so the fate of a cell is what it will become if development is correct, and an embryonic fate map is a representation of all the cells in the embryo that indicates what they will become. Okay, there's another term, potential, I used, or potency. So developmental potential is what a cell can become, and so this is slightly different from fate. So a cell at different times has a number of choices it can make, but over time that potential becomes more and more restricted. So as time passes, the cell's potential drops until its final fate is determined. Determination. So determination is part of this idea of potential. This is a progressive restriction in potential that accompanies the choice of a fate of a cell. So you may not see any difference kind of morphologically in the cell. It may look like its neighbors, but there's an implication that inside the cell there's a stable change that's somehow restricting its potential, so that it is following a particular path as it moves to its final fate. And then differentiation is this kind of expression of the fate of the cell. So once determined, it implies a non-reversible change in the cell, such that it shows a clear difference from its neighbors. That is, it looks like a neuron or a muscle cell, a kidney cell, a skin cell, etc. Okay, so these are terms that we can bear in mind. Okay, so today's lecture is just an overview on the developmental mechanisms. What we're trying to think about is how we generate an organism from a single cell. The nuance here is that we should kind of say this in a different way. The egg is a single cell that will give rise, so the zygote will give rise to an organism. The zygote contains, so all the cells have the same genome, they all have the same information, and yet they form an organism. It forms in the correct pattern at the right time. So how do cells know what they should become? The simple way to think of this is thinking about developmental programs. Okay, so even though the cell contains all of the information necessary to make the final organism, it's not necessarily deployed at the same time. So I brought up this analogy of origami here. Okay, does anyone want to hazard a guess why this is up here? No?
Because from a paper, how it got developed into animal, like origami.
No.
So there are these axes. Oh, So there are these axes where the paper is getting folded. So maybe that is correlating to the various axes which are there in the development.
Yeah, that's a good one. Next.
So there is a lot of movement of the page towards different positions in the location wise. There's a lot of movement of that page, and which gives rise to all of these different forms like the head and the limbs and everything. So there's basically the paper that's moving around and having a different fleet.
Okay, that sounds good. And Adita, you speaking as well. Sorry. Let's hear.
Every step is important in origami as well as in the development of an embryo.
Okay.
So if we miss any step, we will not have our final product. So every step is important.
Okay. Almost. Yeah.
So I was thinking right at the beginning of the first step, the paper is still kind of like a representation for like a predetermined, it is full of potential in a way, can become anything at this point. And then as we go along the steps, they become more and more constrained in what form it can take.
Okay.
That's again like a representation of development as it progresses.
Yeah, fine. Anjum, you're still unmuted. Do you want to say something?
I was thinking the basic thing means the paper itself is there, so the genome of the cell is completely there. It is the steps in which the proteins are expressed, the folds that we make that give us the final organism.
Yeah, that's good. Anindita, you were going to say something. Do you want to?
I was just referring to the different stages involved to yield the output. I think Bhavesh
Okay.
brought it up. Yeah.
Okay, fine. So what it basically is, I have no idea how to use this. Okay. So I'm trying to use my mouse. Okay, so what it essentially is so all of you are right to some extent. So developmental programs are generative. The information is deployed at specific times, and so here on step 1 to 2, the information is to fold. So here, let's step back. All the information to make this bear in step 21 is contained in the paper in step 1. Okay, but to get from 1 to 21, you need to go through all of these intermediate steps. So you're all correct. You're deploying a program in step 2 to fold the paper in half. You're deploying a program from step 12 to step 13, for example, to generate some kind of rudimentary head. Even within the origami, the folds that are making the head are slightly separate from the folds that are making the body and the legs. And again, this is very akin to the way development works, where you have this hierarchy of instruction that restricts going down into particular parts of the organism. So you're all correct. Here we're saying that developmental programs are generative rather than descriptive. Okay. With this, at the top you have a dog oocyte, and here you have a final dog. Okay. And so how all of this happens is kind of fascinating, and is something that we're going to try and explore today. Okay. So with this, you see probably the first issue that we're going to talk about. This dog, my dog, contains many cells. The zygote here is divided once, only has one cell. So you can see this here. You can see this process going on here in C. elegans. You can see the division happening in different planes. You're getting division. The division is somewhat coordinated sometimes. You start getting division in particular areas which are coordinated. And then you see morphogenesis happening, and very soon gastrulation, and then the coalescence, the morphogenesis to give the final form of this roundworm. So I should say that because developmental biology is kind of depends on time, there's going to be a lot of movies, which sometimes seem relevant and sometimes don't. So that was the kind of outside of the C. elegans. Here you can see the division happening. So these are embryos that are labeled, so the nucleus is red and the spindles are green. And you can see here this process as well. Okay. So one of the first things I want to convey to you, and it's probably the easiest thing, so let's just get that out of the way, is that from one cell comes many. Okay. So cell division is a thing. Okay. All of you know mitosis, I think. Yeah. Anyone need a reminder? Good. Mitosis is only a very, can you hear me? Am I?
Yes.
Okay,
Yeah.
good. Okay. So mitosis is only a very small part of the cell cycle, okay, as you know. The cell cycle consists of M phase, G1 phase, S phase, and G2 phase, okay? M phase is where the cell is dividing. This goes on to a G1 phase where the cell grows. When it gets to a particular size, you initiate S phase. S phase is where you get replication of DNA, replication of organelles like mitochondria. The G2 phase is kind of the quality check as a cell prepares for division, and then you get M phase again. So all of these various phases are controlled by cyclins and cyclin-dependent kinases. So everyone is aware of these, are you? Is anyone not? Okay, great. So these cyclins give us an insight into, so they act as ways of, so they act to transition between the different phases. Between the phases there are checkpoints. And so these checkpoints then can become ways of controlling the cell cycle, okay? And cell cycle control is one way in which division and the speed of the cell cycle can be regulated. And so these are some of the ways that either the cyclins or the cyclin-dependent kinases can be controlled. You will not be tested on this, but you can read about it. So there are inhibitors of cyclins, inhibitors of the cyclin-dependent kinases, ways of causing either cyclin or cyclin-dependent kinases to be degraded, so that the checkpoints here that you're seeing can be used to freeze the cell in a particular stage of the cell cycle. Beyond this as well, you have branching of the G1 phase into what's called the G0. Okay, and so G0 is essentially the cell is removing itself from the cell cycle, and differentiated cells are said to be in G0. Okay, so how do we control then the cell cycle? Well, we can use various extrinsic and intrinsic signals. So some of the intrinsic signals are being shown here to act on the components that transition the cell into the various phases of the cell cycle. Here we're thinking about extrinsic signals, and so these are secreted factors that act on neighboring cells. You have mitogens that stimulate cell division, and these can be either by relieving negative controls, or they can be positively transitioning the cell between the different phases. And shown in the figure is a way that mitogen activity could work. It could be a extrinsic signal that acts on its receptor, initiates a downstream cascade of signal transduction, and causes the activation of a particular gene, which enables various activities then to be expressed. You also have growth factors. So growth factors stimulate cell growth by promoting protein synthesis or inhibiting its degradation. Sometimes growth factors are used interchangeably with mitogens. And you also have survival factors that stop cell death. And when we start talking about developmental neurobiology, these survival factors will take on a particularly pertinent meaning. Okay, so cell death by apoptosis is sometimes triggered when the cell is in an unfavorable environment, or when it makes a cue that's it senses a cue that tells it to undergo cell death. And survival factors enable the cell to bypass these apoptotic signals. All four classes of signals can be used interchangeably. Some of the proteins even show a mix of all activities, and this depends on the cell type and the context. I'm going to talk a little bit more about cell cycle in the next lecture when we talk about fertilization and cleavage. And so I think I'm going to leave it here at that. But just to show another movie about some of the problems that we have to deal with when we're thinking about controlling cell cycle. And so what you're looking at is the Drosophila embryo. And at this stage, the Drosophila embryo is just a bag of nuclei. And these nuclei have been, so the DNA is labeled with a fusion protein that labels all the DNA, and you're seeing it as the DNA condenses and then divides. And what you're seeing here is that despite all of these cells being individual cells, at this stage, division is incredibly well coordinated. All of these nuclei are dividing at the same time. And it's like a beautiful dance. I think we have one more division. Maybe not. Okay, so, but as well as this synchronization, you also get desynchronization. And so this is the zebrafish embryo you can see dividing. And now some of these nuclei are dividing, some are not. All of the cells have started doing something different. So this kind of coordination and sometimes desynchronization is important, definitely is important for kind of understanding how regional differences can be set up in the embryo. Okay, are there any questions up until now?
I had a question.
Yeah.
With the coordinated cell division which we are observing, can we attribute some of it to cell memory? Like the cell has somehow an innate memory where it knows that I have to coordinate my division.
Yeah, it could be. It could be. There are many cases where that happens. I mean, definitely when we think about some of the nicest examples are when people are deriving iPS cells, and so these are cells which are forced to become pluripotent, and these cells also seem to retain some of their memory. It's not really clear how that memory is retained, and even in development, there's kind of these positional cues that are set up from a little bit before that will impact what they do much later. And this could be, we can talk about this, we will talk about this in later lectures, okay? This kind of idea of cell memory, okay?
Okay, thank you.
Okay.
I had another question.
Okay.
Where you were explaining the origami part, you said that development is a generative and not a descriptive process.
Yeah.
So I couldn't understand. I mean, what would a descriptive process be like? I couldn't understand that.
Yeah. It's a good question, and it's a difficult question. So a descriptive process is basically saying that here's all the instructions you need. Yeah, get on with it. So it's kind of the difference between having a genome that has all of the instructions within the genome, and then deploying those instructions at specific times. Okay, and so it's a slightly nuanced take on the genome, yeah, and you know it encompasses then. We know that DNA will give RNA, and which gives protein. So it encompasses the generative process encompasses the fact that gene expression needs to be controlled at specific times to develop these particular instruction events in a developmental program. Yeah, whereas a descriptive process encapsulates just the DNA, just the genome, without any reference to its need to be, without any reference for its expression to be controlled. Does that
Yes,
somehow
yes,
answer
it
the
clears
question?
my doubt. Yes,
Yeah.
thank you very much.
Okay. So I mean, that was a great way. Thank you, Kanika. That's a great way of getting to the next point of this lecture, which is to understand how you go from a bag of cells to something patterned. Okay, so here you're looking at my idea of a bag of cells, a bunch of round circles that I've drawn. And what I want to understand is how this round ball of cells will become something more specialized. Yes, Bhavnesh, do you have a question, Bhavnesh?
Nea, ਹੁਣ ਵੀ ਨਾ ਕਰ ਦੋ ਜੇ
Bhavnesh, do you have a question?
No, sir.
Okay, if not, then you can mute. Good. So how do we go from a bag of cells to something specialized? The first thing we need to do is break symmetry. Okay, and so this is a very, very important step in the initial points of pattern formation. The early embryo is symmetrical, maybe, and we need to somehow break that symmetry. By breaking symmetry, we can start setting up various coordinates. And so what I'm going to talk about for the next 5-10 minutes is the amphibian embryo. So the amphibian embryo, let's just go straight to the amphibian embryo, and then I'll come back to this. So the amphibian embryo, the classic one used at the moment is Xenopus. This is this ugly creature on the right-hand side. If my video is on the right-hand side, that's not me, okay? I'm maybe the ugly creature, but it's the amphibian, the Xenopus frog here. And you can see its eggs on the right-hand side are the eggs. The left-hand side is the frog. The eggs you immediately notice are pigmented on the top and unpigmented at the bottom. So they're laid with a polarity already existing that's been imparted by the mother. Okay. Xenopus is a classic model system used for embryology for a long, long time. And the reason why it's so classically used, does anyone want to guess why amphibians have been used so well for developmental studies?
So their larvae are more or less transparent, so a lot of changes can be seen happening inside the
Yeah, so some of them, yeah, these ones, these amphibians are opaque, and most are. So fish embryos are definitely transparent, but the amphibian embryos are filled with yolk and are not. Now there's a much simpler reason, a much more historical and easy reason for this. Thank you, Kanika, for trying, or for
Is it smaller life cycle?
no. Yeah, that's a good guess as well. Reyes?
Isolation of the eggs is easy.
Yeah, that's a good. No, it's a good. That's a good guess as
The
well.
number of progeny is high.
Yeah, definitely. There's a ton of eggs that are laid.
Progeny.
Yeah, there's a ton of eggs laid every time.
And so they have a very elaborate this metamorphosis process also.
Yeah, that's getting it more complicated now. Okay, let me tell you.
Because of external fertilization?
External fertilization is one, but not quite. Okay.
Does it have anything to do with pigment on these eggs?
Does it have any what?
Anything to do with the pigmentation, because it looks like it's half one color.
No. So I'll tell you. Okay, go on. One last guess.
Maybe due to the evolutionary perspective.
No.
Can I try also once?
What were you going to say, Nishan?
I think it's just they had a lot of blooms of these eggs near where they were studying it.
Yeah, that's almost correct. Yeah, and I'll tell you why. Okay, I'll tell you why there are. Okay, so frogs were used as kind of a early pregnancy testing kit. Okay, and so if you injected the frog where the leg met the body with the urine of a pregnant mother, the next day the frogs would be induced to lay a lot of eggs, and so this was used, I guess, in the 18th, 19th century. People then were left with a lot of eggs, and they started trying to play with them, and this became then a developmental biology system. Okay, so the reason why amphibians have been used is, like I think all of you said, like so that the embryos are available externally, so they're externally fertilized, but the availability of embryos was a byproduct of this kind of early pregnancy testing kit, the frog. So you see that there's kind of this difference in pigment between the top and the bottom. And this is reflected in an existing polarity already, okay? And so what the mother's done is to provide one polarity, okay? And here we're showing the polarity from top to bottom. The bottom has some blue cells, and these are emitting a gradient that goes to the top, okay? Now, if we could put in another polarity, we could start getting a coordinate axis. And so here we can, if we somehow were able to label or to assign one cell so that it's red, that red cell could, for example, emit some signal which lays down a gradient. And if we can overlay that with our blue gradient as well, we have now a kind of coordinate system. And this may be a good way of using, of imparting some level of asymmetry and some pattern to the embryo, okay? Of course, this coordinate system is like our longitude and latitude, and these slides were given for high school students. And so that's why Bangalore is labeled there, but I don't think, so a lot of you are not in Bangalore at the moment, so I apologize for that. So the Xenopus embryo does something cool. There's something called cortical rotation that happens in Xenopus embryo. So what you're looking at the leftmost panel is sorry, I'm not able to move one of these windows that I have open. What you're looking at, the leftmost panel, I think, is the Xenopus embryo just at the point of fertilization, okay? And you can see at the bottom there are these green cells. You can see this sperm entry point. Is that all visible? I'm not able to see that panel. Yeah.
Yes, it's visible.
Okay, that's good. Okay, and so soon after fertilization, what happens is the cortex of the embryo rotates in relation to the middle. Okay, and that rotation is dependent on the aster that the sperm nucleus or the sperm provides. This new asymmetry activates these future dorsal cells. So the dorsal means the back, the place where the vertebrae are. It specifies these cells to become dorsal. And this is because now the green cells are in a new relationship with determinants that are in the middle of the embryo, which activate this patterning mechanism. And this patterning mechanism now activates, now specifies this as dorsal, okay? Is that kind of clear so far? Yeah, cortical rotation, yeah. Go on, Rob. Yeah, okay. What's so special about this, okay? And so here, what we're gonna do is talk about the experiments of, so this kind of symmetry breaking happens in many other systems as well. The worm has these pre-existing asymmetries that are imparted from the mother during oogenesis. And so what I'll talk about next week is kind of the importance of setting up oocytes and the oocyte maturation process in many animals, okay? And so the worm imparts some of these polarities already in the oocyte before fertilization, as does the sea urchin embryo here, the second row, as does the Xenopus amphibian embryo here in the third row. One asymmetry how the mammalian asymmetry is generated is still mysterious. And this is in the bottom row here. And I have this reference to this paper at the bottom. I'll put this up on the reading list later today. And so you can kind of understand the issues when it comes to understanding symmetry breaking in mammals. So now that we have kind of some level of symmetry breaking, what I want to talk about now is kind of the experiments of Spemann and Mangold. Okay, so I think I don't know what many of you are taught at university and during masters. Does anyone know these organizer experiments?
So it's basic. So we know about, when we see fate maps, we know like which part of the embryo is going to become what, but we
Okay.
do not know like what really drives that particular portion to develop into whatever it's supposed to develop.
Okay.
So Spemann conducted these experiments on newts. When he did a transplantation at the early gastrula stage, there was no change in the embryo, in the recipient embryo. But when he did it in the late gastrula, there was a change.
Okay. Okay. And thanks, Shubha. Do you know were you told what that change is?
For example, if a piece of new ectoderm was transplanted into the new ectoderm, the ectoderm cell, if it was the late gastrula stage, would develop as the ectoderm cell itself because it was already differentiated into giving rise to the ectoderm.
Okay. Okay. So this is a slightly this one is the experiment that actually Spemann and Mangold were famous for. Okay, and so Spemann, I think you all know. This kind of rather severe-looking gentleman was working in Freiburg in Germany for a long time. Hilda Mangold was his student. She had been a student for 4 years in the lab, and then unfortunately passed away in 1924. 1924, yeah. But not before doing this classic experiment. And so her experiment kind of determined that there's bits of the embryo that have the ability to alter the fates of their surrounding tissue, okay? And what she did was to take the equivalent of the red cell in the amphibian embryo and move it, okay? So the equivalent of the red cell in the amphibian embryo is this structure here that's called the dorsal blastopore lip, okay? And I'll talk a little bit about that in a later lecture. It's essentially where gastrulation starts in the amphibian embryo. It will give rise to the head and the back of the embryo. So it'll give rise to the head, the neural tube, and the vertebrae of the embryo. Okay, and what she decided to do, without really telling Spemann as well, is to bring this red cell and put it on the opposite side of the embryo. And so now she wanted to generate an embryo that had 2 red cells. So here she thought that this red cell was emitting a signal which told the embryo that it was going to become dorsal. And she wanted to put another red cell here to ask whether you would now get 2 signals that make 2 dorsals, okay? And as you can probably guess, her experiment was very successful. And so what she did was to here take the blastopore lip of a host embryo, transplant it into the other side of so the blastopore lip of a donor embryo, implant it onto the host embryo in place of its normal ventral organizer, and ask what you get, and you get a 2-headed embryo. That's cool, and that's very nice. But the important piece of information here, because she had used hosts and donors with different levels of pigmentation, she could tell that the host, that the donor blastopore lip was able to make the surrounding host tissue change its fate as well, okay? And so here, this is a kind of modern reworking of this experiment. In C, you can see, well, in D, you can see the two-headed embryo. In C, you can see a section through the two-headed embryo. The normal notochord, which is a dorsal derived structure, is unpigmented, and you can see that in the section in C. You can see this round circle. You can see two round circles. One is the host notochord, and next to that, this red circle is the donor. Okay. Even though the donor is in red, it's causing the host tissue to form a neural tube. Okay, and so this gave us the idea that that there's conversations going on between different structures to change their fate and change their pattern. So this is communication, okay? And so communication is this ability of, kind of is encompassed in this experiment. Here the donor tissue is telling host tissue that it needs to change fate, okay? And so this process is called induction, okay? So cell-cell communication is necessary to coordinate coordinate information, to coordinate behavior across an embryo. Sorry, I'm gonna stop sharing really quick so I can get rid of this. Play. Yeah, cell communication is necessary to coordinate interactions across a multicellular organism. The ability of one cell to affect the behavior of a neighboring cell is called induction. Okay, these behaviors can include anything. Okay, they can include, as we've just shown, a change of fate. They can be a change in shape, mitotic rate, metabolic activity, motility, any kind of behavior that you can think of. If it's affected by a neighboring cell or even a cell at some distance, it's called induction. Induction can be permissive, okay? And so this can be purely as a checkpoint signal. It just allows the developmental program to take place, or it can be instructive. And here the idea is that without the developmental signal being there, you would have one developmental program. When the instructive induction signal is there, it changes the developmental program to something else. As well as induction, you have something called competence as well. And so competence is the ability of cells to be receptive to these inducing signals, okay? And remember, cells can be induced to be competent to another signal, okay? And so these kind of ideas of induction and competence are incredibly important, and we'll talk about them more in the next lecture. Okay, so before we move on, can anyone hazard a guess as to how induction would work? What kind of signals are we thinking about? Hello?
Mostly external signals, like you showed a slide about mitogens.
Yeah.
So could be through that.
Yeah, yeah. And so if we're thinking about fate change, how would they act? How would fate changes be manifest?
They could be influenced by the environment as well.
Yeah. Okay.
So like if there's a nutrient deficiency, so some factors would signal cells conveying the environmental distress within the cell and then alter fate.
Yeah, could be. Yep. Yeah.
Gene regulatory networks, maybe?
Yeah, fantastic. Yeah. Thank you. That's exactly what I was going to Sure. Georgette, you're ready to speak? You're
Yeah,
right?
I was just going to say that the mitogenic gradients that are usually formed that could also influence cell fate decisions.
Yeah, absolutely. So you bring up a good point, which is that. So I'm thinking very simplistically in terms of, you know, as Anandita said, secreted proteins acting on, as Anthony said, the gene networks. But you're right, Yoja. It could be mitogenic gradients. It could be mechanical gradients as well. So it could be that cells are packed, which are causing fate changes in other cells. And so, I mean, it becomes a semantic discussion whether that is still induction or not. I would say it's induction, but maybe other people would not. So as you already indicated, one way that these induction events could happen is by factor signaling. And so here's an idealized growth factor that induces a phosphorylation cascade and turns on gene transcription and gene networks. So this is a very easy way of thinking about induction, but when we start thinking about gene networks, there are kind of different different nuances and different kind of complexities that we have to think about, and this kind of feeds into the idea of robustness as well that we were talking about last week. And so I put up a discussion forum on the Moodle page, and there's the first paper is on robustness, and so when you have time, just have a look at that, and we'll come back to that kind of issue in about 3 or 4 lectures time. Okay, so this is kind of a straightforward cells talking to each other, and we're going to talk about cell-cell interactions and cell signaling in 2 lectures time. But last lecture we someone had asked me about single cells and developmental biology. Who was that? Who asked me about that? Was that Nishant? I think Nishant had asked me about development of single cells. Is that right?
Maybe it was me. Hi.
Yeah. Hi, Madita. Okay, yes.
Yeah.
Okay, thank you. So I thought I would show you single cells developing. Okay, and this is dictyostelium. Okay, and dictyostelium is generally single-celled, but in times of starvation it will come together. Okay, and so these are single cells moving around, and they somehow in times of starvation, and they'll clump together, and they'll clump together to form a fruiting body. And so this is so there's some level of differentiation in this kind of now new multicellular organism, and you can see that these kind of clumps happening on one side there, and all the unicellular dictyostels streaming onto what's going to become this stalk. Okay, this stalk can move around, so it can go and start finding food, and then it will also and it's called a slug at that point. So you can see it starting to move around here, and it then is just taking kind of this coherent exploring behavior, and at some point it will also sporulate as well. So it will form you'll get differentiation in what is essentially a unicellular organism. It's not quite right, I know, it's not unicellular differentiation. It's become multicellular before it's differentiating. But I wanted to use this as a way of understanding signaling as well, to let you know that signaling doesn't have to be that straightforward cascade that I showed, that it can be something much more simplistic. So it's known that these kind of behaviors are dependent on the ability to sense cyclic AMP. And so the next slide is a different prep. Sorry, Nivedita, you're going to ask a question.
Yeah. So the previous video that you showed, so
Uh-huh.
there the unicellular organisms are basically, so the organism is basically changing between two forms of, two different forms. One is the unicellular form and the other is the multicellular form.
Yeah.
So doesn't that raise questions about what really constitutes an organism? Is it the multicellular being, or is are those single cells can also they also be considered to be individual organisms because they're ultimately constituting one whole in later phases?
Yeah, that's a fascinating question. I think it's philosophical. Yeah, and so yeah, maybe yeah, at some point we can talk about this. I'll as we maybe in the cell signaling lecture we can come back to this, or even after this lecture we can. I think we'll have time. Okay.
It's
But
kind of like that that Portuguese man of war, the jellyfish, which is actually a colony of organisms that acts as a one whole.
yeah. I mean, even you could argue that ants ants act as a colonial organism as well. So yeah. Okay. So here, what we're looking at, or what we're going to look at, is the same dictyostelium organisms with a cyclic AMP sensor. So here they're grown at a larger, at a more confluent activity. Yeah, and you can start seeing that there's signaling happening. Okay, and you're seeing kind of cyclic AMP activity, which then you can start seeing these waves. Can you see those waves?
Yes.
Yeah.
And so these waves precede this kind of aggregation of these dictyostelium unicellular organisms. And you can see them now, kind of these beautiful spirals kind of resolving into these aggregates of dictyostelium. Okay. I think they don't do. Oh yeah. Okay. Sorry. So they're going to aggregate a little bit more. So now these are blazing with cyclic AMP as they aggregate and form the slugs to crawl off and look for food.
So I have a question.
Yeah.
So here we can see the cyclic MP leads to the formation of that aggregate. So does the gradient of the cyclic MP, I mean, lead to the formation of this, or the cells, every cell can sense the same amount of cyclic MP? And
Let's. I'll show you the next video, yeah, and I'll use that to answer. Okay. So here, this is also a slightly better resolution. Okay. You can see these waves. Okay, and so these are cyclic MP kind of waves that are being sensed with a kind of very nice physics here. Okay, and I'll bring up the physics next. Okay. And so you can see this wave absolutely amazingly making these spirals. And this is kind of an example of a reaction diffusion mechanism. Okay. The point I wanted to make here is that the ability of these of dictyostelium to sense cyclic AMP is dependent on just pure physics. Okay. That this kind of pattern of spirals is predicted from a mechanism called the reaction, a kind of a way of understanding chemistry and just general reactions called reaction diffusion, which I will go into in about 3 lectures time. Okay, but this is essentially just pure physics. Okay, these are simple reactions that seem to be trained and drive these complex behaviors. Well, not so complex. Meghna.
is there any similarity between these unicellular colony forming organisms and the multicellular colony forming organisms
Not really.
in their behavior?
Yeah, not really, but the same kind of, you could see these spirals here, yeah? And the spirals are basically activity of cyclic AMP, okay? And you can see the spirals here. And so these spirals are set up by a coupled reaction. And these kind of same coupled reactions are working in different parts of during development to stabilize and strengthen these signaling mechanisms that we've already talked about. Okay, and
What is the thing that has been labeled green in the right panel?
so this is also a cyclic AMP sensor. Okay.
Okay.
Yeah.
I had two questions. First is like how like this can be correlated with quorum sensing we see in bacteria.
I don't know. Maybe, but maybe quorum sensing has lots of different ways of doing this. Yeah. I don't think it's quite quorum sensing. Yeah. Yeah.
Okay.
Yeah.
And the second one was like the spirals are being generated in the media, right?
The spirals are being generated by the bacteria. Okay. So oh no, not bacteria, the dictyostelium. Okay.
Yeah. So it's in the media, right? Like the nutrient is being depleted in the media, and that's when they're sending out the camp, and then that is acting as
No.
an extracellular signal to recruit other unicellular organisms, like unicellular dictyostelium, to come and aggregate.
So in the green, I think, is a cyclic AMP sensor that's genetically encoded in the dictyostelium. Okay.
Okay.
So it's within the dictyostelium. Okay.
Oh, okay.
Okay. And so you can see here in the bright field image here that fairly coherent at this point. Okay. They're fairly confluent. Okay. And so they kind of so cyclic AMP is very diffusible as well. And so this sensor is kind of you can think of it as very freely diffusible. Okay.
Okay.
Pratiksha, you also had a question, did you?
No, sir. No, sir. I am having connectivity issues actually.
Okay.
Sorry.
Okay. So these spirals are interesting, and the spirals are actually pure physics, as I told you. They're kind of they can be described by reaction diffusion mechanisms. And one way to think of this is kind of this reaction here. I can't even say this. Belosov Zabotinsky reaction. Anyone has better Russian than I do? Belosov. Let's call it the BZ reaction, okay? And this is and I can't remember the chemicals in here, okay? But it's simply this kind of spontaneous formation of these this pigmentation, okay? I think this is an iodine salt, and you can see that it's forming these spirals, very similar to what happens in dictyostelium as well, okay? So this is kind of this is pattern formation. But it's not. It's inorganic. It's a regular chemical reaction, and somehow what development has done, and certainly the development of dictyostelium has done, is to take these kind of regular chemical reactions and train them for signaling purposes. Okay. Okay. So with that, I'm kind of done. Yeah, I'm done. I think I covered everything I want to cover. Are there any questions?
I have a question on induction.
Yeah.
So if every cell, I mean, arises from the same cell, so how does some cells become inducing and they induce the formation of different things
Yeah.
within the same?
Yeah. So those cells will be there are kind of upstream mechanisms that will specify those cells. Yeah. And so we know from the amphibian embryo, cortical rotation sets up the red cell, that dorsal blastopore cell. And so there's always kind of this, it goes back to the generative program for developmental biology. There's an upstream mechanism that will specify those inducing cells, okay? And this will keep happening, okay? So the kind of cells whose fate is affected by the red cell, the dorsal blastopore cell, will also induce locally as well. Okay.
Yeah.
Yeah.
Thank you.
Okay.
Could you also mention how one was red and the other was not? I missed that part.
Right. So I'm calling the dorsal blastoporal cells, the dorsal blastopore cells, red cells, just because in the cartoon that I showed, we're calling the cells that do the induction were the red cell. Okay.
No, even in the real image, I think one was reddish and the other was not.
Yeah. So the non. Yeah. Okay. Let's go back to that. So this image, you mean?
Yes, right.
Yeah. So here, this is oh, now I can point. Okay. So this is the notochord, and so this has come from the host, and this has come from the donor. Okay. This red notochord is coming from the donor here. So the donor has been pumped full of a tracer. Okay. But in Mangold's original experiment, the host was unpigmented and the donor was pigmented. And she could show that this donor was coming from a different structure, different embryo, but also that it was inducing the host tissue to adopt a new fate. Okay, so it was inducing new fate into these. This is a neural tube from the host, and this is the endogenous host neural tube. Okay, is that?
It has been artificially pigmented.
Yeah. So this has been induced artificially because this tissue has been implanted artificially. Okay.
Yeah.
Ron, you have a question?
Yes, sir. In the Spearman and Mangold experiment, even though does the position of the blastopore implant have any significance? Because it appears that they were implanted on opposite sides, but if you look at the final organism, they seem to be on the same side, and the body axis is still maintained.
Yeah. So you're right. The kind of new organizer will induce morphogenetic movements that kind of become so the final form is then it looks as though it's on the same side. There are, if you look at the Spearman experiment earlier, let's see if I can show a different image, a different. So I don't have it with me, I think. If you look at a different representation of that experiment here, you'll see that it's on opposite sides now. Yeah. Can you see that? Yeah. And so I think it's just an aspect of morphogenesis and development that kind of remodels the embryo so that the heads are on the top and the ventral is at the bottom. Okay. Yeah.
So it's able to survive even if we do change the axis.
Yeah, I don't know how long it survives though. You know, I don't think it undergoes metamorphosis, for example, but I'm not sure.
Okay.
Yeah.
Raj, in that image,
Yeah.
if the host and the donors are on diametrically opposite points, what really determines which is from which position the tail originates?
Yeah, that's a good question. Yeah, I mean, you can see. Sorry, you can't see. I've stopped sharing. You can see here that there seems to be two tails as well. Yeah.
Yeah.
Yeah. And there's some mess in the middle. So there is we'll talk about this, I think, more clearly when we come up to the signaling lecture. There is actually a ventral organizer as well in frogs, which does specify the tail. The way this experiment is done, it can be done in different ways, okay? So the dorsal blastopore here can be implanted here in the ventral side, either by removing the ventral blastopore, ventral organizer, or by just making a slit in the ventral organizer and slotting this in, okay? So here what Eddie de Robertis has done is just stuffed in the dorsal organizer. What Richard Harland had done in the previous representation is to replace the ventral organizer with the blastopore. So this is not answering your question, I think. Yeah, you asked where the tail is coming from, or
Yeah. So
yeah,
because if we have the two on diametrically opposite points,
yeah.
From my point of view, the tail can either be from the right or from the left.
Ah, okay.
So that is what I was wondering, because there is no asymmetry. There is nothing which distinguishes the two sides from each other.
Yeah.
So how does this asymmetry arise?
Yeah. So Nivedita, why don't you hold on to that question for a couple of lectures, okay? Because I think I'll come back to that, okay? Yeah. Because there is a very complicated regulatory network that operates in dorsal ventral patterning in Xenopus, and it'll become kind of obvious where not obvious. There's an answer to your question there, okay? Okay. So, any other questions? Okay, great.
I have one more thing to ask.
Yeah.
Yeah. In one of the movies, you showed the I think the Xenopus division of the Xenopus embryo cells, in which the DNA was labeled in orange and the spindles in green. So as far as I remember, in that image there was something of a red blob on the extreme left-hand side. So I was wondering what that is. Is it the nucleus from any neighboring cell?
Yeah. So let me see what it was. Yeah. So it's a residual follicular cell. Okay. So when the egg is laid, I mean, the oocytes are surrounded by follicular cells from the mother. Okay. And so I think these are follicle cells, and this is a C. elegans embryo. So I should just show it rather than think I'm showing it. This one, yeah.
Yes.
Yeah. Yeah. So these are follicle cells from the oocyte. They're there sometimes and not in others. Yeah. You can see a remnant here, for example, in the light image. Okay. Which is here. Okay. Yeah. Okay. Cool. Okay. Any other questions? then if not, next week we'll talk about fertilization and cleavage before coming back the week after, the lecture after, onto mechanisms of signaling and things like that. Okay. If you have any questions, you can ask them to me or to Varsha.